Hi, in this video we're going to talk about transport generally of vesicles and with a slight emphasis on proteins. We're going to talk about how vesicles move and fuse at new locations. We're going to talk about how proteins specify where they're supposed to go through localization signals. We'll talk about how proteins are transported in and out of the nucleus and how proteins are unfolded and threaded into the mitochondria. In the next video, we'll talk about how proteins get imported to the endoplasmic reticulum and how they're destined for the plasma membrane or other locations inside the cell. And this is a pictorial representation of the problem. Remember, proteins are made in the cytosol and at the rough endoplasmic reticulum, but there are a lot of locations where proteins might need to end up. So the question is, how do they get there? For the cytosol, the answer is rather straightforward. It's that where they're made, and as I mentioned, the nucleus, mitochondria, and endoplasmic reticulum are specific cases, but once a protein makes it into the endoplasmic reticulum, it can then make it to the Golgi, lysosomes, endosomes, or plasma membrane. So first we're going to say some general points about vesicles, and as we saw in one of the very earliest lectures, vesicles fusing, in this case with the plasma membrane, can release contents that's inside the vesicle out to the outside world, and by invagination and endocytosis can import molecules from the outside world into one of the vesicles. But vesicles can fuse with other membranes like Golgi membranes or lysosomal membranes, and so this is a generic process which will occur over and over again. And here it's schematized between two internal compartments, highlighting not only is the composition of the inside of the vesicle being transported, but proteins that are present in the membrane are destined for the target compartment. And I should also add that if there is any uniqueness to the lipid composition, this is a way of bringing new lipids and perhaps of a different chemical type to a target component. So we'll see that there are at least three different mechanisms by which proteins reach their destinations. One, they can be transported through pores, as in going into the nucleus. Second, they can be transported across membranes, and we'll talk in detail about the mitochondrion and endoplasmic reticulum, which, by the way, uses different mechanisms to get the proteins across the membranes. And then again, once it's in the endoplasmic reticulum, transport by vesicles to other organelles, plasma membrane, and the outside world. This slide we'll talk about how the destination is determined for a protein, and then in the subsequent slides we'll talk about how it actually gets there, but what specifies where a protein is supposed to end up. It turns out that in many cases there are a certain number of amino acids that are dedicated as signal sequences to indicate where a protein should appear. They may not otherwise be fundamental to the protein's function. And what this experiment is showing is that if you use the tools of recombinant DNA and molecular biology to cleave off this amino acid sequence from the protein and attach it to this other protein, you can change the destinations here. The protein in the cytosol ends up in the endoplasmic reticulum and vice versa, all because of the possession of the endoplasmic reticulum signaling sequence. And we think of these as just zip codes about where the proteins are supposed to go. Now, you don't have to memorize these, but these are some examples of the signal sequences. One of the better known is the signal sequence for retention in the lumen of the endoplasmic reticulum, which is KDEL in terms of the one-letter codes, and this is occurring at the carboxy terminus of a protein. Between 7 and 9 amino acids specify import of a protein into the nucleus. And a stretch of hydrophobic residues, as we'll see, can indicate presence in a membrane, in this case membrane of the endoplasmic reticulum as an initial point. So we'll begin with discussing proteins getting into the nucleus, and here we see a picture which shows the nuclear pores, and these are very large holes in the organelle, approximately 50 ribosomes, which are each 4 megadaltons. Each ribosome is close to 100 proteins. Then 50 of those side to side could enter through a nuclear pore, which is pretty reasonable considering that very large RNA molecules with many proteins bound to them need to leave the nucleus for translation in the cytosol. And here's some greater detail of the nuclear pore and some of the data here, a side view, and here looking down at the top you can see the radial symmetry around here, and you can also see sort of this hanging basket on the nuclear side of the nuclear lamina, and even some of these cytosolic fibrils sticking way out into the cytosol. One other important detail that's shown is that there is a large amount of disordered loops here in the center. It's not just a big hole, but it is not a structured area either. But again, these will be specific amino acids with specific affinities for various transporter proteins, and the transporter proteins will be carrying specific proteins themselves in and out of the nucleus. Now on this slide we see a protein, a generic protein with a nuclear localization signal, and there's a nuclear transport protein which binds to the nuclear localization signal regardless of the identity of the major part of the protein which possesses the nuclear localization signal, and the transport receptor will interact with these fibrils which extend out into space, giving it a larger area for interaction, increasing the chances of an encounter. And because of affinity between the protein sequences in the pore and the transport receptor, you're going to have directed motion of the transport protein, which is carrying the protein of import, into the nucleus, where it will dissociate, leaving the, let's say it's a transcription factor, in the nucleus where it can perform its function. Now, without cargo and going in the direction, shuttling back out to the cytosol for another round of import, which is not its energetically favored direction, it's going to hydrolyze a GTP molecule to power the transport, completing the cycle out into the cytosol. To get into the mitochondria, there are a number of decisions that have to be made. One of those is, if you're a mitochondrial protein, where should you end up? Should you end up in the outer membrane, the intermembranous space, the inner membrane, or the inner mitochondrial matrix? And the second point, which is just generally challenging, is if you are going to the inner mitochondrial matrix, you have a double problem in terms of you have to cross two membranes to get inside. Again, a signal sequence is a key element for identifying the proteins that are destined for the mitochondria. The signal sequence will bind to a receptor that's bound to the membrane of the mitochondria. Once it possesses cargo, it can diffuse in the plane of the membrane and interact with a translocation pore of the mitochondria, whereas we'll see on the next slide, the protein is actually pulled into the mitochondrial matrix. The places where these translocation pores are special places where the two membranes are particularly close to each other so that it's easier for proteins to cross. This is the figure from a different book talking about the same subject matter, and it shows greater detail. It emphasizes the fact that the unfolding of completely synthesized and folded proteins is an energy requiring process. So these proteins are folded in their final tertiary structure, and then they are unfolded. They'll interact with the receptor. In fact, they're transported by these chaperone proteins, interact with the receptor, and here we see a molecule which is, again, hydrolyzing ATP to pull the protein into the inner mitochondrial matrix. And in this case, the targeting sequence is cleaved off of the protein, having served its function for specifying the destination of the protein. And now the protein will refold and be ready to perform its activity. So that's how proteins get to the inner mitochondrial matrix. What about all the electron transport proteins that we talked about, oxidative phosphorylation, which end up in the inner mitochondrial membrane? These proteins contain multiple sequences that help guide them to the inner mitochondrial membrane. The first is the same signal sequence, which says going to the mitochondria, but then it's followed by another set of 9 to 10 amino acids, which is a hydrophobic patch, and this is called a stop transfer sequence. As soon as this occurs, it's going to pause in the importation. The protease will cleave off the signaling sequence, and now this portion of the protein is embedded in the membrane, and there it will stay. Now, for proteins that cross the membrane multiple times, there can be several stop transfer sequences, and we'll see a similar phenomenon in the next video about the endoplasmic reticulum. If a protein is destined for the inner membrane space, there is a specific protease which will recognize some of the sequences on the portion of the protein and the stop transfer sequence, which will cleave, and now we have a protein that's free in the intermembranous space. Okay, lots of details, but I hope it was clear enough. Thanks for listening.